And for that, I'm really happy to welcome Vadim Nikulin from Leipzig today, who is a neurobiologist and obtained his PhD in neuroscience at the Biomag lab in Helsinki, doing MEG TMS research. And currently, he's a group leader at the Max Planck in Leipzig. He co-founded the Institute for Cognitive Neuroscience in Moscow and was a visiting professor there until 2022, so quite recently. And his research relates to large-scale spatio-temporal neurodynamics, particularly oscillations in relation to sensory motor tasks, to cognition, and to interoception, which was one of the many reasons to invite him today. He also has clinical research, mostly focused on Parkinson's patients and stroke patients. And he is a well-renowned expert in EEG, MEG, local field potential recordings, non-invasive brain stimulation. I guess whatever's your topic, you can talk to Vadim, and he has a thing or two to say. One last thing is that he also develops multivariate algorithms for the analysis of multi-channel EEG, MEG recordings. And as a personal note, I just think it's a great asset to have Vadim at whatever kind of neuroscience conference. So we're really happy to have you, and the floor is yours.
Thank you. Thank you, Daniel. And I'm extremely happy to be here. I'm very thankful to organizers for inviting me and for extreme warm welcoming yesterday in the restaurant where we already started discussing many things. So my topic today will be evoked responses and neural oscillation in the context of cardioception. So now, when we talk about brain-body interactions, of course, oftentimes we imply oscillatory processes, right? That's important. So another important aspect is that we are often talking about bidirectional interactions, right? So it's a lot about oscillation and so on and so forth. So there is this very nice recent review from Tanya and colleagues showing, yes, indeed, how many brain areas, specific areas relate to heartbeat evoked responses, respiratory rhythms, gastric rhythm. However, what I would like to do today, I would like to focus my talk both on educative part, because many of you here are PhD students, you are getting to know the EEG, MEG, neurophysiology, but I will, of course, also show you our newest findings in the context of what I will be presenting. So now, when we talk about brain measures, EEG and MEG, so there are mostly two classes, evoked response and oscillation. So I just would like you to raise hand, people who study evoked responses. Okay, excellent. Oscillations? Okay, together? Okay, much fewer. So now what I would like to do, I would like to show you that this extreme dichotomy, which is embedded during your studies in universities, might be not true, actually. So, and oftentimes, we should rather treat these two phenomena as one and the same phenomena. So what I'm implying is that a lot of evoked response are not just an evoked response. It's not a unitary phenomenon. It's something which actually intrinsically an oscillatory activity, and I would like to provide you some evidence also in the context of cardioception. So now, in order to do so, I would like to establish a common ground for us so that we can talk using common language. So now, the most classic model of evoked responses is so-called additive. There is no name attached to this response because this is like how we classically investigate evoked responses. So basically what this model says that there are ongoing oscillation depicted here in blue. There is a stimulus or movement. It produces a phase lock response, and when you average many trials, you have evoked response. And then, of course, in this scenario, evoked responses and ongoing oscillations are completely two different processes, right? So, however, in 74, Sayers asked, okay, why should it be the case? So, in fact, evoked responses could be a modification of ongoing activity. So what he meant is that if we have ongoing activity and there is a stimulus, then this ongoing activity can undergo a phase reset, okay? And when you average many of these epochs, what you have an evoked response, but now evoked activity and ongoing oscillations are the same process. And believe me, there are now 20 years of research in different labs around the world where people are trying to prove that auditory, visual, some other sensory responses, especially in human EEG, MEG, are generated through additive or phase synchronization, phase reset process. So let's look at the criteria people are using for differentiating between these two models. So first is that, of course, there should be ongoing oscillations which are required for phase reset. So, however, the problem is that sometimes you don't see oscillation in MEG, EEG, while there are oscillations if you would put electrodes into the brain, because there is phase cancellation, and then sometimes you don't see oscillations. You see kind of more or less flatty lines. So therefore, this criteria is not very good. So the second criteria, that there should be phase synchronization after the stimulus. Well, unfortunately, it applies to both additive and phase reset. So again, not a very useful thing. So the final criteria, that there should be increase in signal power, and that's the case which is required for additive mechanisms. However, due to signal-to-noise ratio, we have a problem. Sometimes there is a kind of additive response, but we cannot see it, okay? And if you think about heart-evoked potentials, they are usually 0.2, 0.5 microvolts. They are extremely tiny, right? So therefore, it's very hard to see this additive part. So now, and I would also like to show you that we had a theoretical paper published some time ago where we actually showed that on the basis of macroscopic EEG MEG recordings, it's almost impossible to differentiate additive and phase reset. Why? So let's consider the following. We have oscillation, these are all simulations. We have oscillations, and many neurons generate oscillation, and their phase is uniform. And then there is an additive response, okay? When you average it, you have kind of nothing in baseline, and then you have evoked response. This is additive. But now look at the phase reset. You have kind of, again, randomly distributed phases of oscillations, but then there is a phase reset and there is a specific phase jump here. But now if you look at the first and second trace, they are identical. So if you record EEG, MEG, you would not find a difference between them, right? And only in the third case, when you have a phase locking of oscillation before the stimulus, and then there is a phase reorganization after, then you can make inferences about the phase reset that it's the case. So now let's look actually what we have in terms of heart-evoked potential. So now there is this paper from Park and colleagues where they tried to show that heart-evoked potentials using intracranial recordings could be generated through phase reset. So first of all, they showed a phase concentration using intratrial coherence. But as I told you, unfortunately, this is not a measure which can tell you whether it's additive or phase reset, okay? And then what they showed that there is no correlation between phase synchronization and increase in power. And that's kind of tentatively implies that we are dealing with this kind of third scenario, which is typical for phase reset. However, the problem is the heart-evoked potentials are so tiny that it's very hard to say that there is no increase. So therefore, although there is some evidence that heart-evoked potentials could be generated through phase reset, still, the conclusive evidence at the moment is missing, but yet it's important because this is theta range, right? And Veronica today presented theta oscillation. That could be actually one of the links how heart-evoked potentials could be linked to intrinsic activity in theta range. That's beautiful. But I would like to show that there is yet another mechanism which can bind together ongoing oscillations in the brain and heart-evoked potentials. And this is due to baseline shift index, which we described some time ago, and it's based on two very simple prerequisites. It's very stunning that it has not been described earlier because it's based on two very simple things. One is that we need to have ongoing oscillations which undergo an amplitude modulation by task, stimuli, and that has been shown in thousands of studies. We don't need to prove it, right? Anything you do, you will have modulation of ongoing oscillations. So that's easy, and that has been shown. But the second condition is a little bit more subtle. So why do we think that oscillation should have zero mean? So let's look. If we have two trials in antiphase and they're amplitude modulated, okay? If you average these two trials, there will be nothing left. However, if ongoing oscillations do not have zero mean, then look what happens. If we have just amplitude modulation of oscillation by stimuli, by heartbeat, by respiration, you name it. And then what happens is that the amplitude modulation of ongoing oscillations cannot average out. And actually, there should be an evoked response. It's easy to see here why it happens. If your signal is amplitude modulated cosine plus non-zero term R, then when you average your oscillations, then of course the oscillatory part will be gone, but the AT times R, evoked response, will remain. You can also very easily show it like if you integrate over phases with over trials with different phases, then all oscillatory part will be gone and you will have evoked response. Now look at it. You don't need additive response. You don't need the phase reset. You just have an amplitude modulation of ongoing oscillation, and voila, you have an evoked response, okay? Again, you don't need to think about evoked responses in terms of different entity. It's the same thing as oscillations. So I just briefly like to tell you how we measure it. Oftentimes, it could be done through baseline shift index, where you extract amplitude envelope of oscillations and also low frequency part. And if the regression between them is positive, it means that you have a positive shift of mean of oscillation. If it's negative, then the oscillations are shifted negatively. So I would just briefly like to show you 2-3 studies where how in a big way we confirmed it and how it kind of could be extended to basically all EEG-MEG studies. So first of all, we use a computational modeling using human neocortical neurosolver developed by Stephanie Jones, you probably know. And we show that doesn't matter what parameters you're using in models, concentration of ion channels, the density of receptors, the length of dendrites, you always have non-zero mean alpha oscillations. Okay, it's impossible to have a zero mean. So now basically what it means that in all of your studies, if you have modulation of alpha, there must be an evoked response sitting on the top of it. Heart evoked potential, respiration, you name it, there must be there. So, and now what we also did, we linked two of the most well-studied EEG phenomena, right? It's P300 and alpha. So these are two canonical brain responses, which practically all of us are studying one way or another. Just Maria presented an example of this P3. So now what we did here, we wanted to show a confirmation for 3 very basic predictions. So first of all, if P3 generated by alpha oscillations, then there should be similar temporal course, there should be similar topography, and stunningly, the polarity of evoked response could be predicted from resting state recordings. Because look at this, if your oscillations have negative zero mean, then if they are attenuated by stimulus like a target in P300, then there should be an evoked response, okay? Well, actually, we confirmed all of these 3 in using 2,000 subjects from live data sets. So first of all, strikingly similar time courses, okay? Second of all, strikingly similar topographies for P300 and alpha modulation. Also, if you do source analysis, then you can see that P300 and alpha are generated in similar brain areas, okay? And finally, if you want to predict polarity of P3, and if you measure this baseline shift index, then you can see it's negative over parietal areas. It means when alpha goes down, P3 should go up. And this is indeed what we found in many of our subjects. So if you split subjects into many subgroups of people having strong baseline shift index negative versus less negative, then what you see stunningly that the people with the most negative baseline shift index would have the strongest P3. But look, baseline shift is calculated from resting state, okay? So we are predicting from resting state properties of oscillations what would be the polarity and size of evoked response. And this is indeed what happens with very high significance. We confirm that the polarity of evoked response can also be predicted from this. And just the final example from this, and then I show you how baseline shift index actually can work for heart evoked potentials, is the study where we used, where we investigated the effect of alpha oscillation on generation of visual evoked responses. So many of you, of course, know that alpha is intrinsically an inhibitory process, right? So our first hypothesis was that it should suppress very early evoked responses. It's the earliest evoked response in cortex is C1 response, which you can get if you use a special wedged checkerboard in visual fields. So, but look at this beauty. So if you look at how prestimulus alpha can generate late evoked response, then you can see that strong prestimulus alpha, right, can lead to a very big evoked response. Why? Because if your alpha is having a negative baseline shift index, then the modulation of alpha should produce an evoked response, right? Look, you will have an evoked response. It has nothing to do with evoked response as such. It rather shows you what happens to alpha oscillations, okay? So now, basically, in order to show how this baseline shift mechanism could be applicable to heart evoked potentials, we just started analysis of a huge live data set. At the moment, we analyzed 98 participants. But it's resting state, and for each subject, we have approximately 1,000 heartbeats. So now, okay, this is the heart evoked potentials. You can see also a typical parietal distributions. So, but now what we did the following. We used a machine learning technique which allows you to see not just any changes in alpha oscillation, but we use common spatial pattern techniques which finds a subspace in changes of alpha oscillations, which would show you in which cases alpha is increased compared to pre-stimulus and in which it could be decreased, okay? So first of all, we analyzed alpha oscillations which are increased in response to the heartbeat. So it's called even-related synchronization. So what we did, we identified pattern in CSP analysis, which has this topography typical for heart evoked potential. So now we can use this filter from common spatial patterns, and we can look through this filter at both alpha and heart evoked potentials. This way, we guarantee that the topographies of alpha and heart evoked potentials are actually identical. But now let's look whether their time courses would be similar. And this is indeed what you can see. If this is how alpha is changing with respect to 0, where it's heartbeat, we see this beautiful increase, but we also have a similar shape in heart evoked potentials, okay? But now you can tell me, okay, what happens if alpha actually goes down? For that, we trained another common spatial pattern, which has slightly different topography, but still it's occipital-parietal. And then the interesting thing is you have a very nice ERD, but when you look through this filter at what's happening with heart evoked potentials, you again see a very striking waveform. So what we are saying here that it might be that heart evoked potential, at least to some extent, could be generated through alpha oscillations, through amplitude modulation of alpha oscillation. So now it's not a standalone thing. It could be a result of oscillatory changes. So Veronica again going with respect to how different oscillatory processes could be coupled with respect to cardiac activity. So you don't necessarily need to have a standalone additive response for HEPs. But again, I want to emphasize it's work in progress. Please take it with a grain of salt. So, but now also another very important thing is if these heart evoked potentials are generated through alpha, and if alpha oscillations have non-zero mean, then there is a little bit of a danger for many of us. So if you are doing sorting, if you are sorting your epochs with respect to different levels of alpha, they could be together with this low frequency activity which comes from a co-modulation of oscillation with non-zero mean. And that's one part of the story where you need to be methodologically very careful when you are sorting anything when there are heart evoked potentials present. Because the differences in heart evoked potentials could be not genuine, but due to the result of sorting and low frequency activity in the brain. So I refer you to this very recent preprint which we have, and if you're interested later on, we can discuss it. So, but now, of course, I want to show you that still the heart evoked activity or evoked responses could be used for inferences about the cardioception. So if we think about, for instance, testing brain states, we have basically 3 main classes of how we can do it. First of all, with oscillations. Alpha, beta, you probably heard a lot of talks about how this oscillatory process can be used for this. Then I assume Daniel told you everything there is to know about 1 over f and how 1 over f process in the spectrum could be used for inferring excitability changes in the cortex. But what I would like also to show you that how actually evoked responses could be used as probe to check the cortical excitability along the cardiac cycle. So, but now, of course, when we talk, we've seen already these images today. If we think about classic cardiac pathways and how potentially they can modulate activity, brain activity, is of course the baroreceptors, which are then working through vagus nerve, goes to nucleus of solitary tract, thalamus, and the cortical areas. And of course, like what we were thinking is that many effects cardiac activity can have on central nervous function, and all of these functions like emotion, cognition, sensory processing could be because there are changes in excitability as a result of cardiac activity, right? So now, and of course there is this new beautiful finding showing that directly vascular pressure pulsation can cause an activation in the brain, for instance, in mitral cells in olfactory bulb. And also the cool thing is that it's extremely fast, right? So oftentimes we find heart evoked potential changes with latency of 100, 200 milliseconds. So that could be a way how it can work, right? So now just briefly to tell you that just 2-3 slides about our prior work showing you indeed how the changes in excitability along the cardiac cycle can affect functions which you probably wouldn't think should be affected. So this is a somatosensory study which we did with Ezra al, where you presented a very, very weak tactile stimulus on the finger, and then basically we look what is the probability of you to detect the stimulus depending on the cardiac cycle. And then, of course, what we found here is that the strongest detection of this weak stimuli is happening during the diastole. We interpreted it in terms of predictive coding because, of course, we don't feel only the pulse on the fingertip, we also feel actually the heartbeats, right? And therefore, potentially, to mask it, the brain can attenuate it predictively, right? And that's why during systole, the detection of stimuli is smaller. And then, of course, it's also nicely confirmed with P300 being stronger during diastole than systole. And this beautiful, remarkable finding that if you have stronger heart evoked potentials before the application of stimulus, then you are more likely to miss the stimulus because you are more interoceptively oriented, right? So, but now this predictive coding story, and I assume that Marseille will be talking about this also tomorrow, I would like you to show that our very recent study where we show existence of predictive coding directly without using additional external information or external stimuli like auditory, for instance. And for that, we use patients with extrasystola, with very frequent extrasystolas. So as you know, there are two types. There is a supraventricular and ventricular extrasystola, and extrasystola is a premature heartbeat, right? And this is a very beautiful model of irregularity in brain activity, right? So it kind of, if predictive coding is working for heart rhythm, then each of heartbeat is in a way a little bit predictive, right? It could be predicted, but extrasystola, it's a very unexpected event. And what is especially relevant, not necessarily the extrasystola itself, but the heartbeat immediately after extrasystola. This is where many people believe the sensation of something strange in heartbeat is happening. So probably some of you experienced it. So now, therefore, we hypothesize that heart evoked potential to this heartbeat, which is immediately following the extrasystola, should be stronger. So now, and this is indeed what we observed. If you compare the heartbeat 3 beats before, like extrasystola minus 3 beats, this is kind of a control condition, versus the heartbeat which immediately follows extrasystola, then indeed we see a stronger heart evoked potentials. Although ECG is not really different between these two conditions, so it's not driven by the periphery. It's, as we think, probably relates to predictive coding, similar to mismatch negativity, like when the following stimulus is not matching the previous one. And also we showed in using source modeling that the strongest changes in ES+1 heartbeat are happening in the cingulate cortex and in the parietal cortex. So, but now, okay, we really have to see that indeed there are these excitability changes which many people postulate, but so few actually showed along the cardiac cycle. So how can we do it? If you want to do it in a very good solid way, one of the best ways I'm aware of is to use transcranial magnetic stimulation, right? So transcranial magnetic stimulation is a method which allows you to use a magnetic field which produces an electric field in the brain. As a result of it, there is an activation usually at the bands of axons and also at the terminals of the axons. Okay, you have the strongest activity. So why it's important? Because when you stimulate brain, you actually directly probe the state of the brain. And in order to probe the state, you can use, on the one hand, motor evoked potentials, right? So this is what happens when you stimulate motor cortex and then something goes down through spinal cord and to the muscles. But you can also use transcranial evoked potentials, which are directly recorded at the cortical level, right? So now, for that, we use resting state and we apply TMS pulses at different phases of cardiac rhythm. And then, of course, we use also a sham condition where we use the plastic in between the coil and the head, mimicking completely somatosensory stimulation, auditory stimulation, all other factors, but not really providing the brain stimulation. And the first thing is that, yes, we observe changes in motor evoked potentials. They are not big, but they are significant, meaning that motor evoked potentials are stronger during systole. And I was super, super happy to see this recent preprint from Tani where they confirmed this, so that indeed in systole there is an increase in motor evoked potential. This is very nice to see. So, but now let's look at the most direct measure of cortical excitability, because motor evoked potential can still reflect spinal cord processes. We want to see what happens with transcranially evoked potentials at the cortical level directly. And this is what you can see. 30 milliseconds after the TMS pulse, there is an increase of transcranially evoked potentials right below the coil. And if you look at the control condition, then you don't see much of a changes here, right? So, but the best contrast in this case is, of course, to suppress sham from TMS. And again, you see this beautiful increase in systole, right? So it's kind of complementary findings. Motor evoked potentials are increased in systole, and then directly recorded transcranially evoked potentials are also increased in systole. So now, of course, we also wanted to have a kind of a behavioral evidence that there are changes in excitability and they are behaviorally in some way manifested. So what we did also, our subjects were pressing on a dynamometer, and we looked at what is the force output depending on the timing when they started pressing on the button, okay? So it could be at systole, it could be at diastole. So what we showed that if you start pressing at systole, your motor output is stronger. And importantly, if you measure the alpha suppression in the motor cortex, and usually alpha suppression accompanies all kind of motor activity. If you are doing some tonic activity, you will always have a suppression of alpha oscillation in contralateral hemisphere. So again, nicely, the alpha oscillation were strongly attenuated when you initiate movement during systole compared to diastole. Okay, so now you can tell me, okay, fine, but how to show that it's not only these measures, but also maybe the bit-to-bit intervals is affected if brain is more sensitive to stimulation at systole. And that's indeed what we showed. We showed the lengthening of interbit intervals when we apply TMS during systole, okay? And the final evidence that excitability changes are linked to heart-evoked activity is by showing what happens with heart-evoked potentials right before TMS pulse is applied, right? So now what we showed here, that if you sort motor evoked potentials with respect to strong, weak, then stronger motor evoked potentials would be associated with stronger pre-stimulus heart evoked potentials. And we interpreted in terms of periods of excitability, cortical excitability, where we have, for instance, 1 second of increased excitability, and it goes both for heart evoked potentials and for motor evoked potentials. So now I just showed you 5 complementary features showing that there is a tremendous variability or like changes in excitability which could be accounted by the cardiac rhythm. So now, and in order finally to show you that, okay, all of these changes in sensory motor systems, they are not just something which you can brush away, but they actually translate to high cognitive motor functions. So now what I'm talking about here, your ability to perceive your intention to move. And that will be done in the context of Libet paradigm. Can you please raise hand, how many of you know what Libet paradigm is? Okay, well, that's great. So as you very well know, this paradigm is famous for discussing free will. I'm not going to jump into this, okay? So I'm a neurophysiologist. I have a lot to say about this, but we are not going to jump into this discussion, no. But just we use this paradigm. And the idea here is the following, for those people who don't know it. So subjects are sitting in the chair and they are looking at this clock. And basically they ask to perform a self-paced movement, okay? Whenever they feel like they need to perform a movement with an interval approximately maybe 5 to 10 seconds, okay? But they have to memorize when the intention to move happened. They have to memorize the position of this red dot, okay? So now what happens is that if 0 is when you are performing a movement, let's say pressing a button, then there is a very nice readiness potential which starts 1.5 seconds before you actually do a movement. But the remarkable thing which Libet and others people found is that your timing when you perceived your intention to move happens actually a few hundred milliseconds after the onset of readiness potential. Now that's kind of why there is all this discussion about free will. It looks like you think you started at this point, but brain decided this few hundreds of milliseconds earlier. That means that you are not in charge. You are kind of post factum experiencing what has been decided for you. We are not going into this discussion, We rather wanted to see how cardiac activity relate to these processes, okay? So now what we wanted to do, we wanted to show that, okay, using a very basic premise, so movement intention or perceiving movement intention, it's basically primarily interoceptive process, right? You are not looking at external information, no. You are monitoring your internal state, right? Because of this, because of your monitoring of your internal state, we reasoned that there should be a locking between the movement onset in W condition, and W condition when, this is when you have to report intention to move. But there is also M condition where you have to report just the timing of the movement. So timing of the movement is more about exteroception, but intention to move, that's more about interoception. So that's first hypothesis. And the second is, of course, because we are more internally oriented, that heart evoked potentials should be stronger in W condition compared to M condition. So, and this is indeed both hypotheses, that's what we confirmed. First of all, we observed the locking of button presses only in W condition. So this is a condition when you're internally oriented, and it potentially can indicate that we could be biased by our heart rate, and that heart rhythm gives a little bit of a timing for your ability to detect your internal processing, for detecting your intention to move, okay? So, and the second hypothesis have also been confirmed. So now what we showed that heart evoked potentials are actually stronger in W condition. This is a condition where you have to have a lot of internal monitoring, right? And that's what happens, and the strongest differences between W and M were also in the parietal areas, where this is the area where we have a lot of interaction between exteroception and interoception. So now, to conclude, what I showed you is that there is a unified framework for viewing evoked responses and oscillations, also in the context of interoception. So what I invite you to think critically is this dichotomy about evoked responses and oscillations. Maybe this is not so straightforward and easy. So really think about your evoked responses oftentimes that as if it would be an oscillatory process. And it makes a lot of sense, Ferenka, what you said, and potential explanation for how interactions between peripheral and central measures could be bound together. So now I also showed you that heart cycle is associated with changes in cortical and corticospinal excitability, and to a certain extent, that can underlie many of the functions we associate with cardiac activity. And finally, I just showed you that there is a central monitoring of cardiac activity in the context of predictive coding and motor intention. So now these are colleagues who contributed to this study in different way. And before I conclude, I would like just to show you that we have now an open position at exactly heart-brain interactions at our institute. If you are interested, so please have a look, talk to me. Thank you.
Thank you so much, Vadim. Questions, starting with Andy.
Thanks for the great talk. So I was having a question with your relation between alpha and evoked responses that you talked about at the beginning. So usually what you find when you look at EEG, MEG, you don't find an increase in alpha concurrently with an evoked response, you find an increase in theta. So how does this relate? And when you look in intracranial data, you typically find increases in gamma and decreases in the alpha and beta range during that, so in response to a stimulus. So how does this relate to what you were talking about before?
Yeah, okay. I like your T-shirt. It says alpha, right? So that's very good. So thank you. I mean, first of all, yes, you're right. So ERS, the theta often has ERS. And there are reasons for this. Actually, oftentimes in rhythmic activities, you have more ERD when you have a lot of activity to begin with. Oftentimes, you don't have ongoing alpha, right? You don't have ongoing alpha, and that's why often ERS could be considered also stemming from additive response and adding power from theta oscillations. You know, it's not really theta oscillation. It could be like an evoked response. That's one thing. Another thing is that while you have ERS in theta, you also will always have concomitant changes in alpha. Like even if you say, okay, I have ERS in theta, fine, but you most likely will have attenuation in alpha, okay? And this attenuation in alpha can create an evoked response in the shape of alpha activity changes, which will be riding on the top of your theta changes, okay? So there is no contradiction as such. Then there is more elaborate explanation. I can provide it later, but it really would take me like 2 minutes. But like showing you how ERS can still produce an evoked activity while you don't see much of a pre-stimulus theta, okay? That's also possible.
Good. Martin, next.
Thanks a lot, great talk. I have 2 questions. One is about this explanation of the evoked activity based on the baseline shift. So how do you think that this relates on how do we apply this DC removal on the recordings?
Yeah.
And the reason why I'm asking this, I think, personally, I don't think there is any way out, in the sense that there is an inherent, arbitrarily subjective way in which we can apply this, because it depends on the length of the recordings, right? Or it depends on the way we actually segment the data, the way we apply the DC. And then this baseline shift also it depends on this filter, for which I don't think there is any standard or golden truth for which we can decide in a way or the other.
Yeah.
And the other thing that I would like to ask you, for which I don't have an answer, but I would like to know your opinion, is how much of all these systole-diastole differences depend on how do we define them?
Yeah.
And do this definition change on a trial-by-trial basis? And how much does it depend on also the duration of the systole versus the diastole duration?
Yeah, good, thank you. I will start answering one by one. I detected like 3 or so questions. So first of all, about DC. So DC, direct current, right? So this is something which is constant, the zero term in Fourier analysis, for instance. So it will not affect anything, because when I talk about baseline shift index, I'm talking about changes from one part of the signal to another. So if you remove DC, what will happen? This whole curve will go up or down. But look, the shape of the curve will not change a bit. So what will happen with DC removal? You will just shift this thing up or down, But this curve, mathematically that will be the same. Now, you're asking how about the periods? Basically, the idea of this whole thing is that the evoked response, which is created by the amplitude modulation of non-zero mean oscillations, is exactly following the amplitude envelope of oscillations. So meaning, if you have alpha, alpha is your carrier frequency. So carrier frequency can only be modulated by processes with a considerably lower frequency. Let's say alpha cannot be fully modulated by theta, but let's say, well, by theta, yes, by theta and lower frequencies, theta, delta. So that's why I'm saying that it will also depend on how fast is your rhythm. So we also detected baseline shifts in beta. And beta, of course, could be modulated much faster. So therefore, your question, what is the time segment in which you can see it? It actually directly relates to how fast your amplitude modulation happens. And the prediction from this model is that literally the shape of evoked response literally follows the amplitude modulation of activity. It's basically 2 sides of the same coin, okay? One jumps from another. They are intrinsically coupled. So it's not like you can have a little change between them. No, they are the same thing, okay? So now in your other question, you said what to do with definition of systole diastole, but for which specific study do you mean, or in general?
For your studies or in general?
I mean, yeah.
They change according to the actual shape of the ECG.
Yes.
On a trial-by-trial basis now.
Yes.
So how do we define them? And they also have different duration.
Yeah, absolutely.
So we sample things on a different scale.
Absolutely.
Somehow.
Of course, we take into account all the T waves and so on. So the point is that there are 2 things here to discriminate. One is the circular statistics. They are not dependent on it because it's bit-to-bit interval and it's 2 pi. And that's okay. And a lot of things which we showed actually depends on the locking. So for instance, what we showed with Ezra, what we showed with Libet, what we showed with transcranial potentials. So there is a certain locking, right? So you will have a locking and it will not be affected. So the most important question, does cardiac activity affect your studied phenomena? It's not so much depends on exactly how you define it. It's rather depends on what do we make out of it and how we interpret it, right? In terms of, for instance, baroreceptor activity or this pulsation in the brain, which is detected at a specific timing. So now, oftentimes the effects we find in systole diastole, they are so much shifted in the direction of middle of diastole that little changes of, let's say, 50 milliseconds, they're not so dramatic. And again, it will be more for interpretation. But you're right, we take into account the whole ECG waveform also for this, yeah.
Right. Andrew.
Thanks. So I just had a question about the last couple of slides you presented, which went a bit fast for me, so I might have missed something. You're talking about the intention to act, monitoring the intention to act, which sounded to me more like introspection rather than interoception. It sounds like you're
correct.
You're focusing on an internal state, but it's not really necessarily an internal state relating to the body. So I just wanted to invite you to say maybe a little bit more about that.
Yes.
And whether you see an important distinction there, or whether you want to argue that that should properly be considered in terms of interoceptive processing. And then relatedly, I was thinking, well, it sounds a lot like you're attending to your own intention of when to move, which is related to sort of making an inference about when I should act.
Yes.
And that to me sounds like you're moving away from predictive coding to something more like active inference. And I wondered if you have considered sort of moving in that direction or in some other direction beyond just a purely predictive coding story.
No, but if you noticed, I didn't use the framework of predictive coding for that study. So that was mostly for extrasystola. But no, thank you. You are right. It's, yes, of course, this is not the same thing. But let's put it this way. The ability to detect your intention, right? Would you agree that it's mostly has nothing to do with exteroception, at least? Like, that's why we have W and M condition, right? So M condition is all about you detecting the movement. So, and this is a very important control, right? So this is not just something which relates to cardiac activity per se, or to your attention and so on and so forth. So that's really something drastically different from exteroception and interoception. So now, it's okay, so you agree that this is more oriented internally, but I agree with you to equate this with interoception. This is quite a jump. But anyway, our previous study, as I showed you, the PNAS paper, so actually it looks like indeed if you are looking more toward, if you are more oriented toward interoception, then you are more likely to miss exteroceptive process, right? Like some other sensory impulse. So here the reasoning was that at least your internally oriented intention or attention should be more associated with bodily processes. And this, we are not the first one to postulate this relation. So there is a paper by Olaf Blanca and Park, right, in Nature Communication, showing how intention to move, or no, the movement, voluntary movement, is actually locked to a respiratory cycle, right? So now this is yet another activity. But yes, I like your idea about the active inference. We will think about this. Yeah, thank you.
All right, maybe one final question so that we keep time. Denise.
Maybe something really brief. So we observed this trade-off between exteroception and interoception. But then my question is, could it not be that if it has to do with cortical excitability in general, we might expect an increase in both simultaneously? So why do you think that does not happen, but rather the trade-off sort of?
Can you again repeat it? Because that was...
So if cortical excitability may underlie an increased HEP, let's say, could it not be that also it could underlie an increased exteroception?
Yes.
So in which case we would expect sort of a simultaneous increase in both and not a trade-off model.
Yeah. So let's see. Oh, no. So as I showed you here, right? So if you look at, okay, but here, right? So what I show here is that if you look at heart evoked potentials before the stimulus, right? And to some extent, the TMS could be considered an external stimulus, right? It comes out of the blue in a way. So, and if you have sustained activity, pre-stimulus and post-stimulus, then you will have a co-modulation of both, right? But look at this, there was no attention involved in this paradigm. So people were just at resting state doing nothing. And that's why we observed a co-modulation of heart-evoked potentials and transcranially evoked potentials together, right? So, but if you think about attention, like in case of this somatosensory detection, so then it's something else. There is really, I think there could be a reciprocal relationship between exteroception, and interoception. And I didn't have time to show this, but we, for instance, show that, answering your question specifically, I think the beta oscillations could be one mechanism which could, on a fast scale, modulate interoception and exteroception, because we showed that, if you know probably works of Stephanie Jones, that if you have strong beta oscillation before the stimulus, then you are more likely to miss the stimulus. But at the same time, we showed that if you have stronger beta oscillations, your heart-evoked potentials will be stronger. So I didn't show you this. This is just also work in progress. So therefore, I think it's also not just one rhythm, but a combination of different rhythms. And also, importantly, whether it's active or passive task. So that would be the general answer.
In the interest of time, let's thank Vadim again. Thank you so much.